This meeting is now being recorded.
Hello, everyone. My name's Aidan Flynn. I'm the chair of the meeting today. So I have been working as a statistician in the industry for more than 20 years. 10 of those were at GSK, where I was Director of Biomarkers and Predictive Analytics. And then I moved on and set up my own consultancy business. This is the PSI Pharmaceutical Statistics Journal Club, focusing on biomarkers, and it is sponsored by Wiley. We have two very interesting presentations today. The first one by Tara Lee, and then another presentation by Jing, Hui, Mike, and Sami. So each presentation will last 20 to 25 minutes, after which there will be some time for questions. So we'll move straight into the first presentation by Tara Lee. So Tara Lee Reddy is a senior statistician at the Biostatistics Unit of the South African Medical Research Council. She holds a bachelor's degree in actuarial science, a bachelor's and master's degree in statistics from the University of KwaZulu-Natal, South Africa. In her current position, she serves as co-investigator and lead statistician on HIV, AIDS, and tuberculosis studies, and has contributed to several publications. In 2013, she was awarded a bilateral grant for a doctoral study at the University of Hasselt in Belgium under the supervision of Professor Geert Molenbergs. Her research interests include the design and analysis of HIV, AIDS prevention trials, and methodology for the analysis of complex longitudinal data. So, Tara Lee, over to you. And for those who are not following online, can I remind you to tell everybody which slide that you are on as you go through the presentation?
Yes, thank you, Aidan. Hi, everyone. I'd firstly like to thank the PSI for inviting me to speak and giving me this great opportunity to share my work. The title of my presentation is A Novel Approach to Estimation of the Time to Biomarker Threshold with Applications to HIV. This work is in collaboration with Geert Molenbergs, Edmund Njeru-Njagi, and Mark Arts from iBiostat. So moving to the outline slide, I'll commence with a brief introduction, going through the need for why it was necessary for such methodology to be developed, and a background to investigating HIV progression. I'll then review current approaches to estimating time to threshold, and then we'd go through our proposed approach, followed by an application to a cohort, data emanating from a cohort of HIV-positive individuals from South Africa. And then I'll discuss other applications and further work that is currently underway. Biomarkers for clinical events are particularly useful in the study of HIV due to the long natural history of disease. The CD4 count is one of the two key biomarkers of HIV, and in the absence of clinical manifestations, the initiation of treatment is based entirely on a single CD4 count. Currently, the WHO recommends treatment for all HIV-positive individuals, but states that it should be prioritized for individuals with a CD4 count less than 350 cells. The time to reach a relevant CD4 count is used as a surrogate endpoint in studies examining HIV progression. However, CD4 count is subject to a high degree of fluctuation and measurement error, Many of the reasons cited in the literature for its fluctuation include that of stress, diurnal variation, and even the menstrual cycle. Hence, a single CD4 count below a relevant threshold should be interpreted with caution. This is where the whole idea about applying a persistence criteria or two consecutive rules came in. And the convention in this approach is that one would only conclude that true disease progression has taken place when two consecutive low measurements have been observed. So here we first extract the time to event, which is then analyzed in a second stage within the survival analysis framework. Now moving to the next slide, some of the issues with this current approach is that it assumes that these event times are observed without error, yet they're based on an error-prone marker. It's also not viable when the interval between visits is large, and patients who enter the study with a CD4 count already below the threshold of interest are generally omitted, which can seriously bias results. Hence, there's the need for a method which takes into account the underlying marker trajectory, measurement error, and left censoring. Next slide. There are two key model-based approaches. One is that of multi-state models, where you would partition the continuous biomarker into a few discrete states. Some issues that have arisen here is that quite often you cannot simply estimate the time to reaching a certain state, as reverse transitions are often encountered due to the high degree of fluctuation. Another approach is that of inverse prediction, where you first extract the true patient-specific marker trajectory by using a linear model possibly, and then you would solve for time. The issues here is that it's quite easy to see that you cannot accommodate complex functions of time. And in the classical framework, the properties of this PI are quite difficult to compute, and simulation might be necessary. Moving to slide 4, we propose a two-staged approach. In the first stage, a mixed model is fitted to the longitudinal measurements, resulting in patient-specific predictive values, which are a function of fixed effects and empirical base estimates. In the second stage, the probability of experiencing two consecutive measurements less than a relevant threshold K at each time point is computed. Using these estimates, the time to obtain two consecutive low measurements is then computed. Moving to slide 5, we'll just go through the methodology underlying this approach. So letting Yij denote the CD4 count observed on individual I at time point J, where J equals 1 corresponds to some time origin at which we start considering the individual as possibly zero converting. The time to event Pi can then be expressed as the minimum or first time at which the previous and current CD4 count measurements were both below threshold K. It then follows that we can present the expression for the expected time to threshold as outlined in equation 2, where we use Sij to denote the probability of an individual experiencing the event or stopping at time point Tij. Next slide. We specify a linear mixed model which satisfies Yi equals Xi beta plus Zi bi plus epsilon I. So here we assume that the random effects and residuals are independent, and beta and bi represent the fixed effects and random effects respectively. It then follows that conditional on the random effects, the outcome vector is normally distributed with mean vector Xi beta plus Zi bi and variance covariance matrix sigma I. Assuming conditional independence in this linear mixed model such that residuals are uncorrelated, these joint probabilities which form Sij simply reduce to the product of individual probabilities. We now introduce a new notation of Cij minus 3, which denotes the continuation probability and can also be interpreted in the survival framework as an individual still being at risk for experiencing the event of two consecutive low measurements at time point J minus 3. So the last three terms in the sequence are confined to be a high measurement followed by a previous low measurement and a current low measurement, as you can see. Next slide. It also follows that we can further simplify things by using the distribution we just stated. Now, these cumulative normal probabilities can be expressed as a simple function of the standard univariate.
All guests have been muted. All guests have been unmuted.
By subtracting the mean and dividing by the standard deviation. We further found a recursive relationship of the continuation probabilities. So when I first presented the expression for the expected time to threshold, it would be clear that as the number of time points increase, the number of ways in which the event could be experienced or could not have been experienced increases. And this can be quite computationally intensive. But when you look quite closely, you'll see the number of ways of continuation of series follows a recursive relationship, and that follows a Fibonacci sequence, and that led us to derive this recursive relationship, which is found in more detail in the appendix. So this greatly simplifies things and means that we merely have to calculate the first three continuation probabilities by hand, essentially, and afterwards we can just use this recursion to get the ones thereafter. Moving to slide 8, and looking at estimation and inference, we propose a conditional version of the non-parametric case bootstrap to compute 95% confidence intervals. In the first step, individual I is removed from the full data set, and in the second step, we sample with replacement from the remaining n-1 subjects. In the third step, the data of individual I is appended to the bootstrap sample, and in the fourth step, we compute the time to threshold. This process is then repeated 1,000 times, and the percentiles of the bootstrap distribution is used to form the confidence interval. Next slide. We now move to an application, and this application is based on the Sinukutemba cohort, which is a Zulu word for give hope. And this is a cohort of 336 HIV-1 subtype C chronically infected adults enrolled at the McCord Hospital in South Africa. And the study took place between August 2003 and 2008. CD4 counts and viral loads were measured every 3 and 6 months respectively, and the ARV guidelines, which were in effect at the time of the study, recommended that treatment be commenced when the CD4 count reaches 200 or below. The median CD4 count at enrollment was 357 cells per millimeter cubed, and the mean viral load was 4.7 log copies per mole. Moving to slide 11, you'll see that the profiles for 8 selected subjects are depicted. And from this, a few features become apparent. One is that there's a high degree of both within and between subject variability. And this is presented on the square root scale for CD4 counts. You'll also note that patients drop out at various times of the study. So these will be touched on in a bit more detail in the slides to come. Moving to slide 12 and returning to our two-stage approach, in the first stage we fit a linear mixed model to the CD4 count evolution. So we investigated baseline covariates as possibly explaining the CD4 count trajectory, and the one that stood out was that of viral load. So categorizing viral load as being low if it was less than 15,000, medium if it was greater than between 15,000 and 100,000, and high if it was greater than 100,000. We fitted the linear mixed model, and we found that individuals who entered the study, I'll just put my cursor on here, with a high viral load exhibited a much more rapid rate of decline in CD4 count compared to individuals who entered the study with a low viral load. And there were 2 time origins that we investigated. And that the first was that using enrollment as the origin, and the other time scale was using the date of 1st of August 2003 as the origin. And the time scale that provided the best fit to the data and also facilitated our interpretations at a later point was that of using enrollment as the origin. So all further analysis would be conducted on that particular time scale. On the next slide, slide 13, I just go through a few key assumptions. So given that these patients, several of them have entered the study with a CD4 count already below 350, for example, it's crucial that we create some kind of window so that we can back calculate when they may have first attained these 2 consecutive low measurements. So we allow a 10-year window relative to enrollment where we consider an individual as having the potential to have experienced the threshold. And the discrete times which fall outside this observation period were created in accordance with the study design of 3 monthly visits. The series was truncated at the visit at which the predicted value dropped to 0, and the origin first time was defined as the minimum time at which the predicted value was less than 1,500 cells. This is based on the maximum CD4 count observed in an HIV-positive individual. Moving to slide 14, we've decided to draw attention to 4 specific profiles, and these are 4 subjects, patients A, B, C, and D. Patient A is one who started with a CD4 count close to 800 and maintained a CD4 count greater than 350 throughout the period considered, so for the full 5 years. And if you move over to the 2 little figures on the right, you'll see the first one represents the probability of experiencing a CD4 count less than 200. And for this particular patient A, this probability was 0 up to even 10 years after enrollment. However, the probability of them reaching a CD4 count less than 350 tends to slightly increase after 5 years, as you can see from the curve on the far right. If we look at patient C, for example, this is a patient who would have experienced a CD4 count less than 350 far before enrollment, possibly. And this is depicted from the curve on the far right, where we see that their probability of experiencing a CD4 count less than 350 tends to increase at 5 years prior to enrollment. Moving to slide 15, where we present the second stage, which is where the estimated times to threshold are computed for all these individuals. And you'll notice here that we've examined both the thresholds as our previous slide, and the threshold of 200 and 350. So what is clear is for patient C, for example, in orange, the patient only actually had 3 visits. And it's quite clear that the confidence intervals for the expected time to threshold are considerably wider for this individual, given that the number of observations were quite few. So for patient C, who entered the study with a CD4 count of 204, they would be expected to first attain 2 consecutive low measurements, less than 200, about 0.38 years into the study, and they're expected to have attained 2 consecutive measurements less than 350, more than 3 years prior to enrollment. Moving to the next slide. Some of our key findings, one was that we had 30 individuals who had a 0 probability of obtaining a CD4 count less than 200 throughout the period considered. And when we delved a bit deeper, we found that these individuals had a protective HLA allele, and they were considered elite controllers. So this just adds supporting evidence to the fact that there are elite controllers or long-term non-progressors in the community. Excluding these individuals, the percentiles of these estimated times were computed. And we found that 15% of these patients had already attained 2 consecutive measurements less than 200 more than 6 months prior to first presentation or enrollment in the clinic. And considering that the guidelines were that treatment initiation should occur at CD4 counts less than 200 at that time, this just draws attention to the late presentation for testing that is happening. We also have noticed that 35% of the patients had already attained measurements less than 350 cells more than 2 years prior to enrollment, which also, since guidelines had subsequently changed to be raised at a threshold of 350, this was also quite alarming and draws attention to the late first presentation to the clinic for HIV testing. Moving to slide 17, since we've created these individuals, these windows of observation for individuals who had already entered the study with a low CD4 count, it's crucial that some sensitivity analysis is conducted to look at the effects of introducing these. So scenario 1 is the base scenario, and scenario 2 looks at truncating the series at 5 years as opposed to 10 years. And scenario 3 and 4 look at whether visits outside of the observation period occurring at possibly irregular time points has some effect on the estimated time to threshold. Scenarios 5 and 6 look at the effect of missed visits outside of the period of observation and whether these could have had an effect on the estimated time to threshold. These results are presented on the next slide. So here in the table we have for each of these participants the estimated time to threshold under each scenario. So for patient A, who's a patient who had a very slow decline, you'll notice that truncation of the series under scenario 2 can have a substantial effect on the estimates. So caution does need to be exercised for participants who exhibit very slow decline with a slope close to 0. We found that results were quite robust to changes in the actual timing of visits considered, and this is clear from scenarios 3 and 4, where results did not vary considerably. We did find that in individuals who entered the study already having attained a low threshold or with a value less than the threshold of interest, that missed visits can potentially have an effect on the estimated time to threshold. So particularly if you examine scenario 6 for the patients C and D, you would be getting a value for patient D, for example. Their base value is minus 0.20 relative to enrollment, but under scenario 6, where visits are missed, they would attain a value of 0.02. However, we noticed that the methodology tended to be robust for the general patient, so the patient who it does not have a slope that is close to 0, or who enters the study above the threshold. So it is important to take these things into account when performing the analysis. In the next slide, we just discuss, and in the paper it's discussed in quite a lot of detail, other applications. So diabetes is one application where confirmatory testing is required, and where 2 consecutive measurements of the HbA1c greater than 6.5% is indicative of diabetes diagnosis. And in prostate cancer, we use the PSA as a marker of recurrence after surgery. And also in aortic aneurysm studies, we look at whether the aneurysm exceeds a diameter of 55, and normally that is when some surgical intervention is put into place. On the next slide, I'll just present some conclusions and further work. So the methodology we've proposed is flexible in that it can accommodate flexible functions of time. We could even look at generalized additive mixed models, splines. So it's quite flexible, unlike the inverse prediction framework. and also it's quite computationally efficient, particularly due to the recursive relationships that we found. There is some additional sensitivity analysis which is required, and in the analysis we've presented, we've assumed that dropout of the study was based only on observed measurements, and it would be interesting to investigate what effect it would have if the dropout of the data was possibly missing, not at random. It would also be of interest to extend these results to overcome the somewhat restrictive assumption of conditional independence and accommodate correlated residuals, and we've made some considerable progress with that. And in some studies, a 2 consecutive rule is studies actually go beyond that and use a 3 consecutive rule. One is in ARV adherence, where missing 3 consecutive doses is considered, leads to the patient being considered as non-adherent. So it would be very interesting to look at different stopping rules as well. And on the last slide, we just have a few key references. So thank you for your attention.
Thank you, Tara Lee, for a fascinating presentation. We're now going to start the Q&A. If anybody wants to ask a question, you will need to unmute your phone, and you can do that by dialing *6. And after you ask the question, if you can mute your phone again. I guess whilst people are preparing their questions, Tarly, I'm interested in, what in HIV, what is the impact of late presentation or delayed treatment, and would that impact really drive the use of this methodology?
Yeah, I mean, the impact on universal test and treat, I mean, if patients are actually first presenting at such a late and advanced stage of the virus, it actually means that test and treat, universal test and treat cannot really work unless we somehow increase our education programs to get people to first test earlier. So this could be useful in predicting how long before or how long patients wait until they're at a more advanced stage of disease before actually going into the clinic and testing. So it could be quite potentially useful there, and to look at predictors of late diagnosis. So, and the hardest thing that people have found is how to define late diagnosis. So this could greatly assist in defining late diagnosis.
Yep, indeed. I mean, you mentioned a potential application to diabetes. I know one big problem with diabetes and late diagnosis is the potential, to have retinopathy and ultimately blindness. So there's a potential big impact there.
Yeah.
Okay. Any questions from others on the line? So *6 to unmute.
I've got a question about dropout. I wondered if, from your familiarity with the HIV data set, what are the reasons for the dropout, and do you think that missing at random could be a concern?
So based on our analysis and on our work with this cohort, participants were removed from the study at the time at which they initiated ARVs, and this was based on their observed CD4 count measurement. So if their measurements dropped to less than 200, or depending on the time at which the ARV guidelines that were in play, this decision was based on the observed measurement. So it seemed that this was plausible for those particular cases. We do know of a few cases where participants moved away, and so they were not removed from the study and due to their actual measurement, and that can be considered to be missing completely at random, possibly. But we have not actually looked at, there were 8% of cases where we did not have a reason for why they didn't return, and we were unable to track those patients. So it is possible in those cases that they may have, succumbed to a more severe disease severity, and it would be good to look at the effect in those cases.
Thank you.
Okay, any other questions from people online? I have another question in that case, Tara Lee. Has there been any work done just on the trajectory of CD4 cells and how it relates to disease progression? The reason I ask is obviously you're making an assumption about some linear relationship, but perhaps there are other models which actually look at the modeling the actual biology.
Yes, indeed. That's actually a very good question. So it's found that in cases where in acute infection, actually the trajectory is actually not linear. And in our particular case, we have chronically infected individuals who are entering at various points of the disease. So we did investigate many different models before coming down to the linear model providing the best fit. We did investigate cubic splines as well, and a linear model in this particular case provided the best fit. But I do think that if it was in an acutely infected cohort, the situation would be different, because they're clearly in the first few months after infection, they're clearly different. The trajectory does differ at certain change points across time.
Okay. And have you applied the model to, you did mention other potential applications, have you applied it elsewhere?
Yes, so we currently have some work under review which looked at abdominal aortic aneurysms and looking at the time to reach a diameter greater than 55. And we've also looked at viral load, which is a marker of treatment efficacy in HIV-positive individuals. And according to the definition of treatment failure, it's when 2 consecutive viral loads greater than 1,000 are observed. So we were able to use some of the methodology developed here, but it did present an additional complication since there are limits of detection. So taking that left censoring into account was a bit difficult, but that's an application that we've also done, and it was quite an interesting one.
Very good. Okay, so any other questions from those online? Otherwise, I'm going to keep hogging the floor. Okay, Charlie, back to me again. So obviously, there's an assumption of conditional independence in your model, and have you tried to relax that or look at the sensitivity around that, and what have you found?
Yes, indeed. So it's something that we worked on quite a lot, and we were able to extend the methodology to incorporate serial correlation. And for this particular data set, we looked at an autoregressive correlation structure, and we used the assumption of the Markov property, so that conditional on previous measurements, future measurement is only dependent on the most recent measurement. So that way we were able to get away with going as far as bivariate probabilities throughout. And so it did make things a bit more computationally intensive, but it did prove that in cases where the serial correlation is high, and actually where the correlation between measurements is greater than 0.6, you can see a substantial impact in the estimated time to thresholds.
Okay. Okay, and so you have published this research. I assume you have developed some code that will run the models. Is that code available, or is that just something that you use in-house?
No, so it is available. We've made it available upon request as it stands, but we're looking at actually putting it out there and possibly, at the moment it's in SAS code, and we've had a few requests and we've sent through the code, but we're looking at setting up a website and then possibly a library, building an R library.
Okay, excellent. I would be interested in finding out more about that. So I'm going to give one more chance to others to ask a question. No? Okay. Tarly, thank you very much. That was a really interesting conversation and presentation. So now we are going to move on to our next presentation. And we have a group of 4 people involved in this. First, Jing received her PhD in statistics and a master's in epidemiology from Stanford University. She's been working in the biomedical field for over 15 years, and her research interest focuses on statistical methodologies in trial design, genomics, and machine learning. She's currently a VP of bioinformatics at Veracyte, a molecular diagnostics company responsible for designing, implementing, and executing bioinformatics pipelines and statistical analysis across all phases of product development. Then we have Hui Yang, graduated from the University of Rochester. Since then, she's been working as a statistician in Amgen oncology therapeutic area. Her research topic includes adaptive designs, biomarker analysis, model selection, and model averaging. Mike Hale is a VP and Head of Statistics and Programming at Shire. He proposed and developed the current paradigm of PK/PD-based clinical trial simulation in the early 1990s. He's chair of QSPI, which is the Quantitative Sciences in the Pharmaceutical Industry, and a committee of the Society for Clinical Trials. He received a PhD in mathematical statistics from Iowa State University. And finally, Sami Tang, currently Biostatistics Associate Director at Vertex. She has about 10 years experience in bioinformatics and biostatistics. Sami's research interests are primarily in the area of adaptive clinical trial design, biomarker subgroup related statistical issues in precision medicine. Sami graduated from the University of Michigan Technology University with a PhD in statistical genetics. So I believe it's Hui is going to be doing the presentation, and as before, we will take questions at the end of the presentation. So Hui, over to you.
Thanks for the introduction. Can everybody hear me well? Just want to test.
Yep.
Okay, great. First of all, thank you for the opportunities for us to present our work. So in this presentation, I'm going to introduce you a visualization tool that can help you measure the performance of the biomarkers for guiding the treatment decisions. So this work is actually collaborated with Sami, Mike, and Jing. So feel free to chime in if you have any questions or comments, Sami, Mike, and Jing. So next slide is the overview of the presentation. I'm going to start with the motivation and background, and then introduce you the tool that was called the weighted predictiveness curve. In the third section, I'm going to show you how to implement these visualization tools in R, and actually the package is already built in R. and then following that will be the conclusion. So nowadays, it's common that in the clinical practice to see with the same therapies can have the different impact across the whole patient population. And most of cancer treatments can only benefit to a group of the patients that can be administered. So it becomes quite important and to identify the patients who are likely to benefit from this particular treatment. In this way, it can save patients from unnecessary toxicities and also increase their chance to receive the drug that actually can help them. From the payer perspective, it can control the medical costs, and also it improves the successful rate of the clinical drug development. We know that it takes a lot of time and the cost to really get successful of the drug. Ideally, we want to get the drug to the right patients at the right time. Nowadays, more and more drugs has been already approved by the regulatory agencies for the particular subpopulations that were identified by the particular biomarkers. As the biomarker becomes more involved in this clinical trial development and drug development, identifying the roles of these candidate biomarkers to predict the patient outcomes has become quite important. So our tools will help you to have an overlook of these biomarkers in terms of the patient outcomes. Now it's in the slide number 6. Talking about the biomarkers, there are two very important biomarker categories: prognostic biomarker and predictive biomarkers. So in this slide, on the left upper corner, it shows the standard two-arm trials without any biomarker identified. We'll see the control arm versus the treatment. Well, on the left bottom corner, you will see that these patients were identified with the biomarker negative, which is in blue, and the biomarker positive, which is in red. From here, you see that the biomarker, no matter what kind of the treatment you are taking, the biomarker positive also always give you the higher survival probability compared with the biomarker negative group. So this is what we call the prognostic biomarker that measures before the treatment to indicate these long-term outcomes for patients that untreated or receive the standard of treatment. Well, on the right upper corner, you will see for these particular scenarios, with the biomarker negative groups, you don't really see the treatment difference between the standard of care and the treatment, while in the marker positive, you see a huge treatment effect. So this is what we call the predictive biomarker. To successfully identify the predictive biomarker, we can identify the patient with the positive markers to give the treatment, can let those patients have the best outcomes. So the tools is trying to help you to identify the prognostic biomarker and the predictive biomarker, and ideally help you to get the particular treatment for the particular patients. Now we are in slide number 8. It's quite common that in oncology studies, survival data are used very frequently. So to visualize the survival, to visualize the time to event, the Kaplan-Meier curves will used very often. This plot shows a family of the survival curves. So each curve presents the survival rate over the time for the patients who have the certain biomarker values identified with the different colors. But through these Kaplan-Meier curves, we can't really straightforwardly seeing the relationship between the survival rate versus the biomarker. We can only see the survival rate along the time. So because of that, we proposed the weighted predictiveness curve that can visualize the treatment effect by the biomarker values. So for the fixed cutoff time, it can visualize the treatment effect. The survival rate in this situation can be projected as a function of the biomarker, and the relationship can be seen through this graph right away. It can particularly become useful for the predictive biomarkers, where the treatment effect can differ with the different biomarker values, which we call it treatment effect heterogeneity. So this method has been published in this paper, and so we also implement in the R2s, which I will introduce just in a little bit. So talking about this predictiveness curve, we provide two approaches to get this predictiveness curve. So the first approach is when you have more information of the predictiveness, the relationships between the biomarker versus the survival rate, we can implement this parametric predictiveness curve. So this approach will take the biomarker as a single predictor, and we implement the whole data using the Cox proportional hazard model. For a given time t, we can estimate the survival rate using this Cox proportional model. And then we provide the three options to get this confidence band, which is commonly used for the plan and log and log confidence intervals. Well, in most of the scenarios, you don't really have the proportional hazard assumptions, and you don't really know the relationship between the survival rate and the biomarkers. So that's the scenarios when we introduce this non-parametric predictiveness curve. So this slide shows you the standard procedures to generate this non-parametric predictiveness curve. We will start with ordering the subject based on the biomarker values from the small values to the larger biomarker values. And then we identified a series of these overlapping windows by fix the window, the size of the window, and also the window moving forward steps. Here we provide the different options, and for example, we can fix the number of the subject in each window, and also we for the speed we will just fix the number of the subject dropped on the left and added on the right. And then for fix the time point for each window, we will just use the standard approach, the Kaplan-Meier curves, to get the proportional surviving the survival rate for that time and assign that to the median biomarker values. To avoid overfitting, we implement these local regressions to smooth the point estimate within each windows and to get this predictiveness curves. Since this is not really can derive the exact formula, so we use the bootstrap the resampling to get this confidence band for our non-parametric predictiveness curves. So you may get a little confused by what I said now. Now let me show you a graph that can just illustrate this process. Just a little moment to download. Okay. So in this graph, the upper panel is the scalar plot between the overall survival and the biomarker. The dot indicates the death and the circle indicates the censor case. The bottom panel is going to show you the predictiveness curve estimate from above. Can you click it? Yeah, so for the fixed time point, a series of this survival rates for each biomarker values, which showing in the right part, estimate from the series of the highlighted overlapping windows. We specify those windows by fixing the number of the patients in each windows and also the moving steps for the sliding windows. So which is the patients dropped from the left and then added from the right to keep the same number of the patients within the windows. After done, we estimate all those estimates from the windows and use the noise to generate a smooth curve. So for this particular examples, the blue lines shows you the predictiveness curves fitted using the parametric approach, while the red real lines will show you the non-parametric predictiveness curves. And you can see that the overall survival tends to get ensured when the biomarker values is between 60 to 40, while for the Cox parametric predictiveness curve, they lose this 10. So the better part of the non-predictiveness curves is can show you the local change by incorporating this sliding window approach and avoid overfitting by using the smooth technique. So this way, it's different from the traditional Kaplan-Meier curves. You can actually see the survival rate versus the biomarker predictiveness curve. So can we go back to the slide, please? So we have built these tools in the R package called WPC, Weighted Predictiveness Curve. And now I just show you a few functions and how we will implement this method to in our daily work. So here, the first function is called a solo WPC curve that can generate the single curves using the parametric weighted predictiveness curve or the non-parametric, showing the upper panel is the using the parametric approach and the bottom showing the non-parametric approach. And you see with the same case studies, the non-parametric curve shows the local trend that can be captured. And also we can just by implement some parameters, we can get this confidence band. That this is showing you the 95% confidence band for both of the approach. And so for the non-parametric curve, as I mentioned, we provide the different options to identify this a series of the overlapping windows. And the previous example we show you, we will fix the number of the subjects and the biomarker width in terms of the biomarker scale will vary in each biomarker windows. Well, we can also provide you the options of the fixed window biomarker window width in terms of the biomarker scales, and then the number of the subjects dropped into the windows can be different. So these particular options will be particular benefit when you have the uniform distribution of these biomarkers dropped into the different scales. So this graph show you that those are the different results by using the fixed number of the patients in each window, and then show the biomarker scales window width. And also we can provide you the different options to draw the multiple curves. the function called dual WPC curve and also the triple WPC curves. So the upper shows you the different ways to generate those two curves. The first is using the parametric approach, and the next two are using the non-parametric approach with the fixed number of the patients and fix the biomarker window width. Just want to show you that what we want to. Sometimes when, for example, the first plot, we can tell that the blue line shows you the treatment and the red line shows you the standard of care. So in this scenario, the patient will be benefit in the treatment arms for most of the biomarker range. So it will be wise to using this, the patients will identify into this treatment arm. Well, for this group, we will see the cutoff point here. It will be more ideal to subscribe the patients that was their biomarker is underneath the 20 will go to the treatment to the standard of care arm. Well, when the biomarker is larger than 20, they will subscribe into the treatment arms. And also the tools can show you to implement the curve, the three different curves into the graph and to give you a better comparison. Also, other than the different approach to fix the number of the patients or the number of the biomarker width, within each window, we can also assign the different weight functions into to get this survival rate. So the standard way to do it is using the Kaplan-Meier curve that's giving you the uniform weight to get this survival rate for the median of the biomarker values. But if you want to have the more weight to the observations that was closer to the biomarker estimation, you want to assign more weight to the patient that was closer to the estimate point. So in this way, we were using the normal weighted and also the Huber weighted. And this is showing you the result by using the different weighted functions within this windows. So here, just trying to show you the different comparisons by using the different values for each of the parameters. The first one is like we assign the different window width, and the second plot show you the different speed. The third one will show you the different function that can like got the plot, that's the picture plotted. So from the simulation we have done, we find out that the Cox parametric weighted curve is good when the assumptions are strictly needed, and also there's the log linear relationship between the survival rate and the biomarkers. Well, for the non-parametric predictiveness curve, it can be used for most of the scenarios, and it's flexible, and it can get a better result compared with the weighted ones. And in terms of this non-parametric predictiveness curve with the different parameters, the small moving steps and the small window size will work better, and also the more centralized weight will work better, which means the normal weight will work better than the Huber and also than for the uniform. So get back to our conclusion. These tools can provide the flexibilities. It can allow the users to visualize the treatment effect on survival as a function of the biomarkers. It can plot one graph or two lines or the multiple curves at the same time, can provide you some guidance, and you can identify that whether that was a prognostic biomarker or the predictive biomarkers, and allow you to identify whether there's a cutoff point that are particularly interested in the patient population. And it can give you the estimate only, and also you can have the confidence bands by using the different options. It allows you to get the different methods to identify these overlapping windows by fixing the number of the subject and varies the biomarker width, or fix the biomarker width and varies the number of the subjects. It can also allow you to try the different methods to get the estimate survival rate in terms of the window width, the sliding speed, and the way it functions. So the tool, as I mentioned, was built in R, and it can be used. It can compare the treatment effect, and it can detect the high impact biomarkers and help you identify between the prognostic biomarkers versus the predictive biomarkers. And ideally, you can design a biomarker-based treatment regimen through our tools. So, yeah, this is the end of the talk, and thank you for participating.
Hui, thank you very much again for another very interesting presentation. So do we have any questions from others online? And again, I'll remind you, you need to unmute your phone by using *6. While we're waiting, Hui, in the animation that you showed, which actually was really helpful to understand what you were doing, was that biomarker data real or was it simulated? The reason I ask is that it seemed like there was reduced survival for patients with intermediate biomarker values, and quite often we don't think of it in those terms. We think of as the biomarker value increases or decreases, the risk goes the same way. So was that real data or was it simulated?
Yeah, this is just a simulated data. I just want to show you that if there is some local trend that the non-parametric predictiveness curve can identify those local trends, well compared with the parametric ones, it can't really identify those curves. But that's a good point. I can really see the value of the approach, and in particular the non-parametric approach for maybe defining optimal thresholds in biomarker data. Now, for the parametric approach, is there any benefit of using your approach versus, say, just fitting a model where you'd have a main effect for biomarker for a prognostic marker and then an interaction effect with treatment to get a predictive marker? Do you get the same? Would you get the same results? Can you say it again? Sorry, I didn't really quite understand.
Yeah. So is there value in using your parametric approach over, say, just fitting a survival model where you would have a main biomarker effect for a prognostic marker and an interaction effect for a predictive marker?
Right. So for for our predictiveness curves, that we can provide you the trend for the survival rate as a biomarker. So by draw a curves, and if we have the multiple curves, we can actually compare whether there's a cutoff, whether we can identify those cutoffs as the other ways we can't really see those, we can't really identify those, and it becomes more interesting if there are two curves and come cross each other. we can see it through the curves.
Hi, this is Mike speaking. Can you hear me?
Yep.
So, to your question, the model fit requires the premise of model form. The Cox model typically uses where the hazard is a linear function of the biomarker, and the non-parametric approach doesn't require those kind of assumptions.
Okay. Yeah, no, I understood that, Mike. But I was just wondering about the parametric approach.
Yeah.
Hello, this is Jing. Can you hear me?
Yep.
Okay, so I'm thinking like in the linear form, like even in the parametric setting, of course, those most common will be using the linear term, but there's nothing preventing us to adding more interaction terms as well as higher power term like quadratic term. So I believe if it is indeed just, as you say, a linear mean estimate, then probably the two approaches are equivalent. But I believe even under the parametric assumptions, there's still potential generalization opportunities that make this model more flexible.
Yeah. Yeah, and actually, I mean, there's real value in just the visualization part, because I think, the people that we work with, the medics and the clinicians, they would love to see those graphs. So I think there's real value in that alone.
Yeah, I think the original idea when we were doing this is really trying to help the clinician to look at the visualization, to really explore multiple marker or multiple treatment difference within the biomarker. So we have actually discussed, talked about using the model-based approach. The only problem is it's really just estimation per se. It's not giving people more exploration, and especially if you have a multiple biomarker, then your model-based approach has to be more complicated to different parameters to be associated with it.
Also, this is Jane, going back to your earlier question regarding the particular animation we showed, although that is from a simulation, I believe we do have a real-life data where towards the boundary of the biomarker range, we do detect some local trend beyond just monotonical decreasing or increasing. So sometimes I think real-life data do show us more complexity than we initially expected, making the non-parametric approach maybe more favorable in most of the real-life applications.
Yeah. Yeah, and I also think in practice, if we have a continuous biomarker, and I know a lot of our clinical colleagues will automatically start thinking in terms of dichotomizing it in order to make it practically useful. So, do we recruit this patient or not? And that needs to be based on some decision rule. And, your non-parametric approach could really help to inform that. Okay. Sorry, go on.
Yeah, I just want to add, yes, so for that part, yes, that particular cutoff that can really help the inform the team that whether we will approve the patient or not. But yeah, just want to add some comments.
Okay. Okay. Do we have any other questions? Okay, so back to me to take over again. So it is becoming clear, not just from the presentations today, but I'm sure we've all seen it in our day jobs and conferences that we go to, just how important biomarkers are to clinical development and to the future of healthcare. We've heard about precision medicine a lot, And I know in my own career, it's an area that I've worked in a lot, but a lot of talk, and I think we're still really trying to work out the way to do it better. I mean, this is a very kind of general question to the team. The most efficient and appropriate way to evaluate the impact of a biomarker beyond what you've shown today. Do you have any guidance you can give to others online?
I think Mike, Sammy, or Jean, they have more experience. Do you want to comment on that?
Okay, this is Sammy. So, yeah. So basically, I think it's a very good question. The biomarker precision medicine world issues have a lot of things we can be doing, especially if you think about when we are doing oncology. If you have multiple treatment, the treatment placebo, even within treatment, you have a different dose level. Looking for the treatment difference within the biomarker positive group, biomarker negative group, you can use this kind of tool and also use different methodology to really develop what should be the treatment you are choosing for your biomarker design perspective. And also a very important one, I think we also mentioned, we were thinking is during exploratory phase 2 studies in oncology world, you can also have a lot of biomarker you are exploring with a continuous platform. The very important part is how can you figure out what is the most promising biomarker can jump out of your exploratory part of the analysis. And this kind of tool could help. I think from that perspective, this methodology are trying to help overall discussion of the biomarker issues we could encounter during exploratory phase. Of course, during the phase 3 part of phase will be a different story. We might not recommend using this kind of tool, but we should think more about, I always have a question like how we can use adaptive design in the phase 3 perspective. It will be hard, but actually there's a lot of people are looking into it. It will be hard to work with, I think, FDA perspective using adaptive design with a biomarker threshold, try to refining during phase 3 perspective. But during our real-world design issues, we do see a problem like that. If you have a biomarker, if you're really sure, you can do enrichment design or stratification design. But if you're not that sure, you have a threshold problem, how can you utilize the information in the design perspective in phase 3? I think that's kind of my perspective within the biomarker-related issue during design or during exploratory phase. I don't know whether any other team members want to add on anything else.
Yeah, this is Mike speaking. We call this a visualization method, and that's really to, communicate a concept more than a precise number or cut point or something. One of the big challenges is we have such a huge number of biomarkers in some situations. I saw a study protocol last week. They had about 30 biomarkers that were being measured, and the team doesn't know if any of those actually are predictive or prognostic. And it's exploratory. And so a tool like this, I think, can be helpful in just seeing visually if there appears to be something going on or not. Because these survival studies can be so hard to figure out. So for me, this is the kind of area where I think it has potential greatest use is when you've got a bunch of biomarkers and you really are trying to figure out if any of them are worth pursuing.
Yeah, no, I can get that, Mike. I mean, one of the big challenges, particularly if you're maybe towards the biomarker discovery phase rather than biomarker development, is when you look at a large number of biomarkers, there will be some that just look to be quite convincing, and there are often experts in the room who will be able to provide some kind of biological rationale why that biomarker is coming up, and we need to guard against that, because it's always possible to come up with some kind of plausible biomarker that actually has just come up by chance.
Yeah, I think that's a really good point. So besides the methodology, we do need to work together with the clinician and the biologist to make sure the pathway perspective is making sense, not just picking by chance. But the methodology definitely will be very helpful working with the team, the whole cross-functional team together to pick up the right thing.
Yeah.
And this is Jing. I just want to add a very last point, because I think another revolution that happening in parallel is the genomic advance. So coupled with what Mike Hill said very insightfully, we're looking at multiple biomarkers, which can be millions. And in that sense, I will call it genomic intelligence, and machine learning sometimes is needed to build a model, to have a companion diagnostic that is the composite index from complex machine learning models, instead of just one biomarker, that you can take advantage of the whole genome information. And our visualization tool really doesn't have to be on one marker, be it KRAS mutation burden or whatever. It can be on an index as well. It could use your a whole genomic information, your proteomic. It can be a prediction score from a model. Then that way you avoid multiple testing and all of going through just marginal distribution one by one, looking at very small effects, hard to be detected by the right power and sample size. Instead, you have an intelligent design of a composite model to give you the right prediction, and this one could also assist you visualizing the composite index of prediction score.
Again, very good point. I guess you also then start getting into the overfitting problem and how you overcome that. But that's probably a whole other session. Okay. Well, let me just, for one last time, are there any other questions?
I noticed on the chat board, someone asked whether we have a package. We do. So, Hui, do you mind to re-emphasize how people can download that package? Where can they find it?
Yes, sure. It's built in R. So you just go to the R website, and the package called the WPC, Weighted Predictiveness Curve. You can just download in your computer, and there is some test data there, and you can play around with the different functions. And there's real data examples, and each section will just show you how the different function gonna work using those play tools.
Yeah.
Okay. Thank you, and thanks. I missed that. I missed that question on the public chat, so thanks for picking that up. Okay. Well, thanks to all of the speakers. Two very interesting presentations, and I know I learned a lot. So thanks to all for joining. I hope you find this a useful session. A reminder that the PSI Statistics Journal Club, the TCs run quarterly. They are open to both PSI and non-PSI members. The next one is planned for June, and they are also free. And then up on screen now are some upcoming PSI events. There's a free scientific meeting on translational statistics, ideas, evidence, innovation, communication, March 30th at the Amgen site in Uxbridge. Then there's the PSI conference, celebrating 40 years of promoting statistical insight. That's May 14th to the 17th at the Grange Tower Bridge Hotel in London. There are some pre-conference courses on the 14th of May, early phase decision making and trial design, presented by James Matcham and Alan Betting. Sample size, power and adaptive trial design for late stage studies, a practical hands-on workshop. And then there's another training course on sample sizing, June 28th to 29th at the Novotel in Catwick, and that's presented by Stephen Julius. So if you want to learn lots about clinical trial design, there are some very interesting courses coming up. So that's it from me, and unless there are any other final questions, I will bring the session today to a close, and thanks again for joining.
Thank you.
Okay.
Thank you.
Okay, thank you. Bye-bye.